Good day, everyone. Today, I'm going to be talking to you about some of the methods that we use in our laboratory for imaging pathological states in connective tissues. My name is Doug Keene, and my co-author here is Sarah Tufa. Together, we've got about 50 years in experience in connective tissue matrix research and imaging. And we'd like to talk to you then about some of the experiences we've had over that time. This is one of the matrices that we are most interested in. We work with it frequently. It is human skin. And here we see collagen fibrils cut in cross-section. We see the basement membrane, the lamina densa. We see lamina lucida, and we see the epithelial cells here at the top. One of the structures that we're most interested in the last few years, certainly, and certainly since we described these structures back in 86 or 87, are these anchoring fibrils. These are banded fibrils which are made of type 7 collagen. They insert into the lamina densa, they loop around into the dermis, and in so doing, they entrap collagen fibrils. So they essentially are responsible for stapling the dermis to the epidermis. There is a disease in which type 7 collagen is absent or near absent. It's called recessive dystrophic epidermolysis bullosa. And in this disease, you'll see, here's the epithelium, here's the dermis here, but you'll see a lack of anchoring fibrils. They're simply not present. Or if they are present, they're present in a very rudimentary form. Should you rub the skin of a person with recessive dystrophic epidermolysis bullosa, even gently, will form a blister, separation between the epithelium and the dermis at the plane of the anchoring fibrils, just below the basement membrane. Individuals who are somewhat mildly affected may just have a few blisters on their bodies. These individuals likely have a little bit more than 30% normal collagen 7. There's also individuals who are severely affected with less than 30% normal collagen, type 7 collagen, or possibly nil to none. These individuals, their patient life expectancy is reduced to around 30 years. They'll likely succumb to sepsis or squamous cell carcinoma, and their lives are quite painful and quite miserable. So we'd like to help these kids get over this disease. The therapies involved are to somehow get full-length type 7 collagen. The dimer of type 7 collagen is 780 nanometers long. This is a rotary shadowed image of type 7 collagen. The monomer has a NC1 domain with what appear to be little roots, and an overlap of about 60 nanometers here between one dimer and the other, staggered in a anti-parallel fashion. There's a couple of different therapies that we're investigating and evaluating. Some are to correct patient keratinocytes or fibroblasts so that they carry the correct gene to make type 7 collagen, and these are then used as grafts or injectable cells. We could subcutaneously inject normal collagen 7 just underneath the skin and hope that this molecule would be incorporated into the basement membrane to form anchoring fibrils. And then a more systemic treatment would be to intravenously inject normal collagen 7 into the circulatory system so that type 7 collagen would then travel to not only the skin, but the esophagus, the cornea, other tissues which are also affected in severe RDEB. Our portion of the evaluation is typically the transmission electron microscopy. So the way we go about evaluating collagen 7 then, these are the anchoring fibrils again, just to remind you, this is the full-length collagen 7 dimer. We have two antibodies as tools that we use for evaluating the effectiveness of the therapies. One is an antibody that recognizes the NC1 domain, another is the NC2 domain. And as we apply these to tissues then, we expect that the NC1 antibody will localize to the lamina densa, and the NC2 antibody will localize to the top of the arch. antibody that recognizes the type of the arch. If that is present, we then assume that the anchoring fibrils are fully formed. We may get some localization of the NC1 domain, and that would be possibly indicative of just a fragment of the type 7 collagen being incorporated into the tissue, but not a full-length molecule. The typical localization, EM localization procedures that we embark on then are either section surface labeling, in other words, lightly fixing the tissue in small percentages of glutaraldehyde, no osmium tetroxide, dehydrated to 90% ethanol, and then embedded into LR white. And then the epitopes are exposed on the section surface, and antibodies followed by secondary gold is then applied to that section surface. Another mechanism that we use most commonly for skin is to simply diffuse primary and secondary gold conjugates into the tissue. This would be into unfixed tissue. And then once the incorporation is in place for both primary and secondary antibody, we would fix the tissue and embed it into spurs epoxy. This tends to be very effective for the lamina densa, or this dermal epidermal junction, because antibodies penetrate really quite far, as we see in this light micrograph, about 100 microns into the tissue. So the localization is really quite easy to detect, and the fixation is adequate to be able to preserve these anchoring fibrils so that you can actually see the arch and see the localization. I'll point out here that one thing that's very important in this TIM block method is to use 1.4 nanometer secondary conjugate. Now, 1.4 nanometer gold can't be seen in our electron microscope without a gold enhancement protocol, but the 1.4 nanometer penetrates far better than 5, 10, or 15 nanometer gold. So we use the 1.4 nanometer gold exclusively for any M block prefixation protocols that we use these days. This is the protocol that we use for M block immunocytochemistry. It's important to mention that the biopsies are transported to us in serum-free Debecko's culture media. We use that because we feel that the cells are very happy in that media, and unless the transportation takes more than a day or two, bacterial growth is not significant in serum-free media. But for longer trips, we'll use Michel's transport media. That is actually quite good for preserving antigenicity, but it's not quite as good for preserving the cells. And the primary antibody and the 1.4 nanometer secondary gold conjugates are diluted again in serum-free media. We used to use a bench protocol, which basically took us about 4 days to complete. We now use a microwave protocol, which takes us just 5 hours to complete. The gold enhancement protocol takes about 15 minutes, so it doesn't add much time to the protocol. So let's consider a little bit, though, as sort of a side note, the 3% glutaraldehyde step here. Glutaraldehyde, as you know, cross-links protein. It has important functions, but fixatives also coagulate, precipitate the tissue constituents, and they're a major source of artifact. Here in this image of skin, we can tolerate the extraction and precipitation, but I'll show you a few images in a little bit on high pressure frozen skin and how it differs remarkably from what we consider to be good fixation in skin. I'll also mention that additives may be included in the primary fixative, also the secondary fixative, to stabilize specific components. Now, here's a good example in cartilage. Here is cartilage fixed in 3% glutaraldehyde. Here's the electron microscopy. And here you'll see that the cells are shrinking away from the matrix. There's a precipitate on the surface. The edges of the chondrocytes are rather ragged, and you can see that they've pulled away from the matrix. Whereas if you preserve the proteoglycan content, proteoglycans are responsible for retaining water in this tissue. If you retain proteoglycans, you'll see an intimate association between the cell border and the albeit precipitated proteoglycans, yet still retain proteoglycans. So this is a very important technique which we really like for demonstrating, especially light microscopy, also electron microscopy in cartilage. If we then look by an alternative technique, high pressure freezing, and compare it to spurs embedded 3% glutaraldehyde fixed cartilage, you'll see again that the chondrocyte is pulled away from this matrix. Proteoglycans are collapsed on the surface of these collagen fibrils, and it's an overall ragged appearance of the chondrocyte, as opposed to the high pressure freezing, which retains this intimate association of the plasmolella of the chondrocyte with the matrix. The matrix isn't particularly easily studied here because there's so much proteoglycan retained that the collagen fibrils themselves are very difficult to image. But certainly this is what we should think of when we think about the structure of cartilage, not the artifact-filled 3% glutaraldehyde. Also in skin, I mentioned I'd show this image to you earlier. So here is skin fixed in glutaraldehyde, and here is skin following high pressure freezing. So a couple of artifacts become immediately obvious in the chemically fixed material. First of all, the lamina densa. Now, the lamina densa is really just a collection of highly concentrated molecules associated with the plasmalemma of the epithelial cells, which in the chemically fixed material just precipitate as this band we call the lamina densa. The epithelial cell shrinks away from the lamina densa, causing the lamina lucida to appear. And we see no lamina lucida in the high pressure frozen material. But I'll also point out that within the matrix, there's a high concentration of proteoglycan that's been retained, so high that you just simply can't see anchoring fibrils. So anchoring fibrils are what we must see as we evaluate epidermolysis bullosa therapies. And if we can't see them, we can't use this technique. I'll also finally mention that the connective tissue matrix is exceedingly hydrated. It's about 70% water in skin. So in order to turn that water into amorphous non-crystalline ice, which is the objective of high pressure freezing, the high pressure freezing successes are just infrequent. So we certainly can't sacrifice a biopsy to high pressure freezing. Now, as we think about, say, for example, skin biopsies, we typically work with punch biopsies up here in the upper right-hand corner. When a punch biopsy is taken from skin, what you'll see is that the skin retracts a little bit from the cut edge that's created by the biopsy punch, but that the structures within the skin and their relation to other structures in the skin are tolerably preserved. But if we work with something like tendon, for example, the Achilles tendon shown here, if we were to cut this tendon and sample it for electron microscopy, it would just simply roll up into a little ball. So we have to maintain its orientation and its linearity by keeping it attached to the other components within the long bone and leg. So in this case, what we would do is remove the skin, we would fix the intact limb and 1.5% glutaraldehyde, 1.5% formaldehyde, decalcify the bone, fix it again in 3% glutaraldehyde, then osmocate, dehydrate, and embed, and then approach the area of interest with block trimming, a razor blade, and ultramicrotomy. So in that case, the relative positions of tendons shown in dark blue and bones shown in lighter blue, fat deposits and muscle tissue here in the center are maintained quite well. And they're only maintained because they were fixed in situ. If we look at tendon then, one of the things that we're very interested in doing, and this is sort of a method that we've developed over time, but it's more software oriented than it is anything else. But here I'll point out, first of all, that all these fibrils are cut in cross sections. So we have managed to maintain the linearity of the tendon. It did not roll up during preparation. And all these fibrils are cut in cross section. But what our collaborators really want to do is they want to see a field, but then they want to go in at higher magnification to actually examine individual collagen fibrils. And you can do this by taking a montage. So this montage was collected by taking 81 images at 80,000 times magnification, but the lower magnification images is displayed at 11,500. So it allows our collaborators then to do what we call desktop microscopy, where they can simply enlarge an area, a field, and move around in that field, measuring different fibrils, seeing unusual fibril shapes, that sort of thing. Very, very valuable, and it's why a lot of our collaborators keep on coming back to this. I'll also mention regarding, for example, tendon or other collagen fibrils, that when we look at these in sections, in fixed and embedded tissue sections cut at 80 or 60 nanometers, we see difficulties in really resolving the structure of these collagen fibrils in those tissue sections. However, if we just do a simple homogenization procedure, cutting a little bit of tendon out of the tissue, grinding it up with homogenizer in the presence of buffer and ice, importantly ice, we see quite a different field of collagen fibrils. We can understand we can start to understand the organization of the molecules in these tendons when stained by 2% PTA. The resolution is just simply so much greater that I would certainly recommend once in a while looking at the tissues by negative stain. Now here's a good example of something that we wouldn't have been able to detect had we just looked at sections, tissue sections. These are isolated microfibrils. Again, they were isolated with a homogenizer. We did not go through a lot of trouble to isolate by size or anything like that. We knew essentially what these microfibrils looked like by rotary shadowing. And here we see a microfibril in a typical 50 nanometer banded periodicity. But as this group of microfibrils got caught up in some debris at this end and this end and stretched during drying, we could see that the periodicity of these elastin-associated microfibrils increased from 50 nanometers to 125 nanometers. So now we recognize that these microfibrils can stretch, whereas if we looked at them only in tissue sections, we get no idea that these stretch, and consider how remarkably different the structure is after fixation, dehydration, and bedding as compared to negative staining. Really quite a difference. We learned a lot by the negative staining experiments. So let me come back then to immunocytochemistry. I mentioned earlier that for the skin experiments that we do, we typically use the in-block method together with a 1.4 nanometer secondary gold. We get good penetration along the dermal epidermal junction. This is typically how we evaluate those epidermolysis bullosa samples. On the other hand, if we had a tissue which was more difficult to penetrate, so simply can't diffuse antibodies so far into the tissue, then we're going to transfer over our attention over to the surface immunolabeling. So here again, fixation in as high a concentration of glutaraldehyde as you can get away with for the particular antigen you're dealing with. No osmium tetroxide, dehydrated to 90% ethanol, and then embedded in LR white. Then the section is immunolabeled. Here's a good example. This is, I'm showing this image as sort of dual purpose. First, to demonstrate that how long bones grow, because that'll be important as I go into the next part of the talk. So this cartilage template is replaced by this expanding front of bone. It's important that this cartilage template be maintained in its size so that it's not degraded. And if it was degraded without this chondrocyte proliferation and matrix deposition, which is normal, if that doesn't occur, then the growth of the bone will simply stop. The other reason I'm bringing it up is because this is a rather impenetrable region. So if we're interested in looking at cells, for example, at the center of this growth plate, there's really no way to diffuse antibodies into this large tissue space. We really have to expose this area to section surface immunolabeling. There are, this is an image of ruthenium hexamine fixed material. Excuse me. This is ruthenium hexamine trichloride fixed material. cartilage growth plate. These are the chondrocytes. But within this matrix are also proteoglycans. There's a number of different collagens. There's also this cartilage oligomeric matrix protein. Now, this protein we call COMP. It's secreted by these chondrocytes and localizes to the matrix and binds many of these other matrix constituents. In a disease called pseudoachondroplasia, what we see in the chondrocytes is an accumulation of these stacked membranes within the rough endoplasmic reticulum. So we would like to know what is in these accumulations of stacked membranes. But we can't diffuse an antibody into this cell without using Triton or saponin or some other membrane permeabilization procedure, which would destroy the ultrastructure of the cell. So this is a good situation where we would use section surface immunocytochemistry. Now, I'll mention that Peter Dinklage's cartilage looks like this, and it's the reason that his long bones are short-toothed. So if we apply then section surface labeling to localize COMP, this mutated molecule present in pseudoachondroplasia, we see that the mutated molecule is accumulated within these stacked membranes within the rough endoplasmic reticulum. And not only is COMP retained there, but so are many other constituents of the cartilage matrix, including collagen 9, collagen 11, fibromodulin, and calnexin. So what happens in this situation is that the mutant COMP gets stuck. It can't be secreted from the RER. It accumulates there, but as it accumulates there, it also accumulates and binds together type 9, type 11, the other structural macromolecules. So protein synthesis within these chondrocytes ceases and growth is arrested. That is pseudoachondroplasia. We were interested, though, to develop a cell culture model to study pseudoachondroplasia. So in this case, the mutant cartilage oligomeric matrix protein was put together with GFP. It was actually YFP, a GFP derivative, and then embedded in LR white. So this is nice. We can see quite a bit of detail, but we really want to see ultrastructurally where this YFP is localizing to, where the mutant COMP is localizing to. So in order to do that, then we simply overlay the fluorescent image on top of the next serial image taken for TEM. We do this using with Fiji. It's a little complicated when you first start doing it, but it's not all that big of a deal. But what we noticed as we were embarking on these studies was that we would get lots of YFP fluorescence in culture, in the live culture. We would get plenty of fluorescence emission in 1% glutaraldehyde. But as soon as we exposed it to LR white, the fluorescence would diminish to the point that we really couldn't see anything beyond background. After digging around a little bit in the literature, we realized that the intensity of many of these GFP derivatives diminishes below pH 7. And in fact, GFP derivatives can be used as pH detectors of changing pH. We also recognize that the stock LR white that we were trying to embed our tissues in has a pH of about 5. So as we looked at these cultures and tried to understand how we might be able to recover the fluorescence intensity, here is a whole amount of these YFP expressing rat chondrosarcoma cells underneath the cover slip. So they're wet and underneath the cover slip. They're actually in LR white. Now, if we then add just a drop sodium carbonate at pH 11, we see that as that diffusion front goes through and underneath the glass and exposes different pH, a higher pH to these cells, they immediately fluoresce. So what we embarked to do then is to embed our tissues then in LR white, adjusting the pH of LR white to pH 7. We also recognize that pH 7 LR white will polymerize in about 2 hours at 35 degrees centigrade. So 60 degrees centigrade was no longer necessary. We do need to displace oxygen in the polymerization chamber with nitrogen. So here we have liquid nitrogen bubbling off the gaseous nitrogen, or we could fill this chamber with gaseous nitrogen through our valve systems at the top with this vacuum oven. Finally, I'll also mention that dual conjugates carry both a small gold particle and also a fluorescent marker, can be applied as a secondary antibody. So in this case, it's another section surface immunolocalization study, but in this case, we're localizing lysozyme to Panna cells. Now, lysozyme, this particular antibody and this particular antigen, would tolerate relatively high percentages of glutaraldehyde, up to about 1% or so. So the structure is actually quite good here, and we could localize not only at the light microscope level, but also at the electron microscopy level. So it's a very powerful conjugate if you can figure out a way to use it. Now, the protocols that we use are, of course, written up on our experiment books with quite a bit of detail. It's difficult to just type all this up and give it to you in one meeting, but I will tell you that if you go to this edition of Methods in Cell Biology, volume 143, you'll find a chapter in there for the ultrastructural analysis of extracellular matrix. And we have written into this chapter very high detail protocols that you should be able to follow, including where to buy the reagents. So if you're interested in pursuing this, our methods in connective tissue, please find this article, and it's a free download. You don't even have to buy the book or anything. So that concludes my talk. I really do hope to actually see you guys at M&M in Portland next year. Portland is where my home is, so I'll look forward to welcoming you at the front door. Thanks very much for your attention.